Today we're really looking at the last part of protein synthesis, which is translation. So by the end of this, you'll understand how proteins are made. There's quite a bit of review as well, and hopefully you'll be able to get through a lot of the material at the beginning, feeling like it's pretty easy. So to begin, if I've not already collected your packet, I will do that. We're going to go over some big picture stuff and make sure you can contextualize and then talk about protein synthesis. Once you're done, you can go ahead and work on the packet. There's a document on Schoology called Translation, and you can use that to work through the modeling aspect of this lesson. And then finally, for our homework, you'll want to finish up the 13.2 notes. Get your vocab done because there will be a quizzy tomorrow. Okay, so we are looking on your packet. If you go to page 18, you'll notice question number 1 talks about the organelles involved in making proteins. And I'm not going to make this too difficult for you, but I do want to go in some depths. So stay listening and I will let you know when is a good time to pause and write down anything you may need. So a little bit of a review. Actually, right now would be a good time to pause and think about what are the cells that you're looking at here. On the top right, we're looking at a prokaryotic cell. And when we think about prokaryotes, we're thinking organisms that have no membrane-bound organelles. Their DNA is free-floating in the cytoplasm, and the only membrane they have is the cell membrane that surrounds them. And the only organisms that are prokaryotic are going to be bacteria. The two types of cells that you're looking at on the left are both eukaryotic, and they do have membrane-bound organelles, and in fact, a lot of those membrane-bound organelles are going to be really important for not only making the protein, but more importantly, delivering it to where it needs to go. And in terms of eukaryotic cells, we can look at two categories. The single-celled eukaryotic organisms are called protists, and they can include things like the diseases that cause African sleeping sickness and malaria, among others. And then multi-celled organisms are going to be those you're probably the most familiar with, which are fungi, animals, and plants. And just another review here, it's important to remember what all cells have. So all cells have, around their outside, a cell membrane. They all have DNA as their genetic material. And they all have ribosomes, and now we can really put this into context for protein synthesis. So that cell membrane is going to be what separates what's on the inside from what's on the outside. That's that selectively permeable or semi-permeable membrane made out of the phospholipid bilayer with proteins and so forth. So cell membrane is really separating what's on the inside versus what's on the outside. Seems logical that that would be an important piece of all cells. DNA is going to be your genetic information, and it's 2 strands of nucleic acids made out of all your A's, C's, T's, and G's. Those are the nucleotides, the subunits that make up the DNA. And let's really focus on this piece here. What matters the most when it comes to DNA, what makes you different from a mushroom, different from the disease that causes malaria, different from a bacteria, is not the A's, C's, T's, or G's themselves, but the order. Kind of like if you take the alphabet, you can change it around, and in one sequence it becomes a Nicki Minaj song, and in another sequence it becomes Shakespeare. Same letters, but now you're just putting it in different orders. And you're going to use the genetic information, of course, to make proteins, and that's happening on the ribosomes. And because of all of the functions of proteins, it makes sense that all cells would need ribosomes because, well, no proteins means no purpose to DNA, which means no life. Big suck. Okay. Looking at ribosomes here, there's a few ways that we can take a look at them. A simplified model, a 3D model, or we can look at it molecule by molecule. But what's really important that you know is, for starters, there are 2 subunits, the small one and the large one. You can probably guess which one is small and which one is large. Okay, spoiler alert, the little blue one, that's the small ribosomal subunit. you can figure out the rest. Each one of these subunits is made out of the rRNA. Remember that rRNA is ribosomal RNA. So part of the ribosome itself is made of RNA and the rest is made of proteins. And so where do you find ribosomes? Well, if you're looking in prokaryotes, they're always free-floating in the cytoplasm. There's no membrane-bound organelles, and therefore there's nothing that these guys attach to. Instead, when you're looking at eukaryotes, there's two ways you can find these, either on free ribosomes, which means they're just chilling in the cytoplasm, or on the endoplasmic reticulum. So if we take a look back at question number 1, what organelles are involved in making proteins used in eukaryotes? We can look at free ribosomes and bound ribosomes. So they're really the same structure, it's just where they're located. Free ribosomes are going to be located in the cytoplasm, and they're what are making proteins that are going to be used in the cytoplasm. Bound ribosomes are going to either be secreted from the cell or sent off to particular target organelles, like, for example, the proteins that are involved in the electron transport chain in the mitochondria can be made on the ER and then transported to the mitochondria. What's unique about eukaryotic cells, again, is that they have membrane-bound organelles. The endomembrane system is a really important part of these membrane-bound organelles. And so we can start by looking at the nuclear membrane. The nuclear membrane is the innermost part of the endomembrane system, and it is what surrounds the nucleus. And unlike the cell membrane, this actually has really big pores that are big enough for mRNA and ribosomes to be able to be made in the nucleus, but then exported through those pores. In blue, you're taking a look at the endoplasmic reticulum. There's two parts there, the rough and the smooth. We've mostly focused in class on the rough, and that's because, well, that's where the ribosomes are. That's really what's most important. The ribosomes attach there, and the ribosomes themselves are making the protein, but the rough ER is what's going to be modifying them. So again, if you take a look at number 1, you should already have the bound and the free ribosomes as two kind of separate components that are involved in making proteins, but then the endoplasmic reticulum, particularly the RER, is what is going to take the proteins that are produced by the ribosomes and then modify them. And the last organelle that is involved in proteins, in being able to produce the proteins, are going to be the Golgi apparatus or the Golgi bodies. And they're going to take the proteins that are made in the ER and then sort them and package them, kind of tag them for a destination, almost like your UPS store would do. And what they're getting tagged and packaged into are going to be vesicles, which you see in green on the image on the left. And those can end up just being temporary, where they transport materials from point A to point B, or they can stay in the cell for a really long time. For example, lysosomes are vesicles that are made and stay there until they're ready to merge with some other vesicle that has materials that may need to be broken down. Okay, so we're talking about, again, proteins, being able to make the proteins. And if we are talking about being able to make proteins, it's really important that we understand what the structure of that protein is. And proteins are made out of repeating units called amino acids. There are 20 different amino acids, and each amino acid is determined by its R group. That R group just means a variable group. So we see the central carbon here in the center, an amino group, which is the NH2, the carboxylic acid group, which is the COOH. This should have a double bond right there, but my bad. And then the hydrogen down here, but of course the R group is really what's important. Think of this almost like beads on a string. And so all of these amino acids are going to string together, and those R groups are what are going to be involved in either repelling each other or being attracted to each other. If they're attracted, they're going to pull close to each other, and that's going to be really important for folding. Okay, if you take a look again at your lecture notes, if you look at number 2 is another review now at the amino acid structure. So I definitely expect you not only to know the amino acid structure, but the reaction that's involved in making something called a dipeptide, or a 2 amino acid. And that reaction is done via a dehydration synthesis. Another good time would be pausing now and trying to remember exactly what this will look like. Make a little prediction here. Okay, so I'll go ahead and show you that dehydration reaction now. Okay, a new bond is formed between the hydrogen of one of the amino groups of the amino acid on the left and the oxygen of the carboxylic acid group on the right. That means that the H2O is free to move away. That is now oxygen. Your nitrogen and your carbons are not stable because they don't have enough electrons in the valence shell, so they're going to go ahead and make a bond between them. So we now have a dipeptide, 2 amino acids, and proteins are just really, really long strands of amino acids, which we're going to call polypeptides. I didn't write this down, and I should have, because it's going to be a really important vocab term. So kind of to the right side in your notes, I would make a note there that a polypeptide is a string of amino acids. So it's P-O-L-Y, poly, P-E-P-T-I-D-E. And we can look at a polypeptide like this with all those different R groups, but really the important part is the R groups themselves. The carbons and nitrogens and oxygens do play some roles, but not quite as much as the R group. So, and if you remember about protein structure, the really important part is going to be the fact that it folds up, and the way that it folds is going to give it a three-dimensional structure that's important. So, for example, if we have this green amino acid, the R group of that one being attracted to, let's say, this kind of purplish gray color over here of this of this amino acid, then they're going to pull towards each other, for example, looking something like this. Oh, pretty neat, right? So I'll show you a quick video. In this video, we're going to be looking at a polypeptide and how it folds. And so here you can see the polypeptide strung along here, with the R groups coming off the side, some different shapes here. You've got double rings, single rings, some branched pieces, some tiny little pieces, and this is a pretty typical polypeptide with hundreds of amino acids. In fact, the one that I showed you in the PowerPoint was really, really small. It was just easier to make it small, but most proteins in your body can have hundreds or even thousands of amino acids. And again, it's going to be their folding pattern that really makes the difference in how they function. So we can see each of those R groups there. And every amino acid has a couple of different ways of writing it. It's got a long name, usually ending in like INE, so alanine or proline. They also have 3-letter abbreviations and 1-letter abbreviations. In this video, you're just seeing the 1-letter abbreviations that are all the way down. So here's kind of the ball and stick model. Here's that beads on a string model. And then we can also look at a space filling model. So what does it look like in 3 dimensions? Oh, there it goes. So it's folding based on interactions of the R groups. And if you ever see a more globular, almost looks like mashed potatoes that someone just flung, just know that those are usually proteins that have that space filling model. There it is. So again, you can kind of see over here, I should have paused, right? There's kind of like a little space, so maybe that's for binding something. I don't know exactly what this protein is, but you can see that there is a 3-dimensional structure to it. So somehow we have to get from DNA to RNA. We saw how that happens, but now to proteins. And they're different languages altogether. DNA and RNA are both examples of nucleic acids, but now proteins are a completely different class of biomolecules. And so we're going to use some different terms here. We're going to use the terms triplets when referring to anything in DNA, codons in RNA. And so the RNA is read 3 nucleotides or 3 bases or 3 letters, A's, C's, U's, and G's, 3 letters at a time. Each 3 letters in mRNA is going to make 1 codon, and every 1 codon is going to code for a single amino acid. So let's say, for example, I had an mRNA sequence that was 27 base pairs long, that would be 9 codons long, which means that it's coding for a 9 amino acid polypeptide. So the process looks something like this, where we're going to have the mRNA sequence right here. The ribosome is going to move along, and every 3 base pairs is going to be recognized as a codon, and a new amino acid is going to be brought into the growing polypeptide chain. So if you take a look at number 3, you can answer that with a lot less detail than you'll need to know overall. The big idea here is that the ribosomes are reading the mRNA 3 base pairs at a time, or 1 codon at a time, to be able to grow the polypeptide, because each codon codes for a single amino acid. And so the question is, how does the mRNA, or the ribosome, know which amino acid to bring? And the truth is that neither of them do. What makes the process work, what makes it so that each codon is recognized and a particular amino acid is brought to the site, is this molecule here called tRNA, which is a single strand, right? It's continuous. If I was to pull on both ends here and here, I would just get one long strand. If I kind of flattened it, it looks something like this. But there are 2 parts that are going to be really, really important. One of them is the amino acid binding site. So there are a ton of different tRNA molecules, and each one has this binding site up at the end. So up here, it can actually bind to a specific amino acid, which are, again, the monomers of proteins. The other important part of tRNA is a part down here called the anticodon. And in fact, the anticodon is what gives each tRNA its specificity, because each anticodon is complementary to an mRNA codon. For me, I remember anticodon and tRNA. So the anticodon is on the tRNA, and then the mRNA has the codon. There's no T there. Okay. So a quick word about codons and amino acids. So if every codon is 3 nucleotides long, then we can figure out exactly how many different codons there can be. So we can look at, for example, AAA, AAC, AAG, oh my goodness, this would take forever to list them out. And in fact, there's an easier way to do it. We know that every codon is made out of 3 nucleotides, and there are 4 possible nucleotides that can go in each position, either A, C, U, or G in the first position, A, C, U, or G in the second, and so on. And so all we really need to do is multiply those together. So take 4, because it can be A, C, U, or G, times 4 in the second, times 4 in the third spot, which means there are a total of 64 different codons that are possible. Now this is where it gets a little bit tricky, because there's 64 different codons, but in your head, try to remember what I said about how many amino acids there are. So there's only 20. And so 64 codons and only 20 amino acids means that there is a little bit of redundancy. For example, I have 4 different tRNA molecules down here. I know that they're different because they have different anticodons. This one says GGA, this one here is GGG, GGU, and GGC, and they all code for the amino acid called proline. What that means is that for the mRNA, the codon that says CCU, or the one that says CCC, CCA, or CCG, those can all code for the exact same amino acid. So there are 64 different types of tRNA, and some of them code for the same amino acid, but each one has its own anticodon down here at the bottom, and that anticodon is what is going to help determine what amino acid to bring to the growing polypeptide. So you've got the background information, all the information that you need. I would take a second and pause and go through it and make sure that you're really comfortable with certain terms. In particular, make sure you understand the ribosome and the ribosomal subunits, what mRNA is and how it's made, what tRNA is and the two major spots on it, and how mRNA and tRNA bind together. And now we can take a look at translation, the process of actually building our polypeptide. Okay, so we're looking at a YouTube video right here. It's already shown the process of transcribing or making the mRNA. So here's our mRNA right now, and it's left the nucleus and moving through the cytoplasm until it binds to the large and small subunits of a ribosome. And here we have an example of a molecule of tRNA. This is a really nice diagrammatic tRNA. Of course, it doesn't look anything like the actual three-dimensional one. You've got nucleotides here that are shown in different sizes, and that's just so that the anticodon is nice and clear, that is what this piece is. So here's our tRNA model, as well as the amino acid that is bound to it. And so that tRNA is going to enter towards the ribosome and link up with the complementary base pair, or complementary codon. So if we take a look here, you can see that the codon in mRNA is GAA. The anticodon on tRNA is CUU. So G goes with C, and in mRNA, A is going to go with U. So this tRNA with its specific amino acid is attracted because of the shape of these nucleotides. So they're going to continue to come in, and now we're using dehydration synthesis to build these peptide bonds kind of one at a time. And here we've got our growing polypeptide chain that is then exiting out of the ribosome. And then once it moves either into the cytoplasm, it'll spontaneously fold, or it will be helped by enzymes that are in the endoplasmic reticulum, and also get some other things added onto it so that it becomes a functional protein. Isn't it pretty? Okay, so we can go back now and look at the last details here at our in our packet, but it would be a good idea now to take a look at question number 4. Pause and see if you can outline the role of tRNA in protein synthesis. And the last thing we need to be able to do is use what is called a codon chart. And a codon chart just breaks down exactly all of the possible tRNAs and the mRNA codons and determines based on what tRNAs we have in our bodies, and in fact all organisms have the same set of 64. There's very little deviation. So I could take DNA from my body and insert it into a squid, and it would express that gene the same way. That is, it would read the DNA into RNA and use those RNA codons to put together the same polypeptide. And so the codon chart is just a way of that scientists have assembled all of the information of all the different tRNAs that we know. And so it's important to remember that this is a codon chart. That is, if you take a look at the mRNA, these are the mRNA codons that are going to be used to build specific amino acids. So for example, if I was to look at UUU, the anticodon on tRNA would be AAA and would carry LEU, leucine, as the amino acid. But we're just going to look at it from the point of view of the mRNA codon. So for example, let's say our mRNA said CAG. The way that we're going to be able to figure out what amino acid to add is we're going to look first on the left-hand column at the first base of C, and then we're going to find the second base A, and go over to that column. Sorry, this is called a row, this is a column. Anyway, so we've gone from the C over to the A column, and then down until the third base spot right there, the G. And so that means GLN, which is the 3-letter abbreviation for the amino acids that's going to be made by the codon CAG. Another example could be GCU. And before I go through and show you how to do it, maybe go ahead and pause and see if you can do it on your own. So again, we're looking at GCU. So I'm going to start at G, over to C, down to U that's in that particular box. And my amino acid is going to be alanine. One more example here for you, UGA, U, over to G, down to A. And so we see a stop codon. A start codon is any combination that indicates that the ribosome should start coding there, whereas the stop codon is the one that kind of acts like a period at the end of a sentence and tells the ribosome to break apart from the rest. And there's two kind of codon charts that you should be familiar with. Now, I will warn you, yeah, you might see one or the other, but make sure you know how to use both of them. This chart is definitely easier for a lot of students, but do not just assume that the first three letters are necessarily the three-letter combination that is the correct abbreviation. So if I take a look at CAG, notice that I'm going to go start from the middle, C, out to A, that's in that quadrant, out to G, and I've got glutamine. Now, a lot of these circular charts show the amino acids on the outside, and they put the entire word there. And if we take a look back at what we just looked at, CAG still codes for glutamine, right? They're all the same codon chart, but the abbreviation GLN is different than you might expect just from looking at the first three letters. So I just want to kind of put that warning out there. But regardless, we can do the same process with all of these. CAG is going to give us glutamine. If we look at GCU, we're going to go G to C to U and get alanine. And notice that whether it's GCU, GCA, GCG, GCC, all of these are alanine because of that redundancy, because 4 different tRNAs all can carry the amino acid called alanine. And then finally, U to the G to the A is our stop, which is a great time to go ahead and stop this little bit of a lecture here for you. So now that we've gone through the PowerPoint and the lesson, if you've got questions, certainly ask me. At this point, you should move on to the translation activity. If you go into Schoology, there's a document called Translation. Follow the directions. Please, please, please read through it line item by line item. Make sure that after you've completed the activity itself, you print the document and put it into page 20 in your packet. And then there's some more directions there, for example, for exactly what you need to label on that document so that you can get full points. And then you can also go ahead and work on the vocab. In terms of notes, you can work on your 13.2 and your vocab should probably be finished for tomorrow. We will have more time tomorrow to finish the activity as well as go over the transcription and translation practice worksheet that is on page 21, and you will have a quizzy tomorrow. You're also going to have some work over break, so if you know that you're not going to be here tomorrow, you'll definitely want to get in touch with me so that we can get you all set up there. So anyway, like I said, if you've got questions, let me know. Otherwise, have a good day.